Thanks for joining us on another exciting journey into the world of science, right here on the SAN Science Experience. If you enjoy this kind of content, hit like and subscribe. It's really the best way to support the channel and keep these episodes coming. Today, we're plunging into a truly fascinating area. This surprising link between how quickly our cells replace their proteins, that's protein turnover, and, well, how long different animals actually live.
Yeah, it's a great topic. And we'll be drawing our insights from a really compelling recent scientific study. This research took a wide-ranging look, a really wide look, at protein turnover in all sorts of mammals.
Yeah.
From the short-lived mouse.
Right, lives just a few years.
All the way up to the majestic bowhead whale.
Yeah.
An animal that can live for, 2 centuries?
Maybe
more.
Exactly. And the central question we're tackling today is, what can the pace, the speed at which our cells rebuild themselves, their protein machinery, what can that tell us about aging and longevity? Why do some creatures that seem to have a slower, maybe more leisurely rate of doing this, why do they live so much longer?
Well, the scientists behind this study, they use some pretty sophisticated methods, cutting-edge techniques really, to measure these replacement rates for different proteins inside the cells across these species.
It's like getting under the hood at a molecular level.
Exactly. It's a real deep dive into the molecular engines keeping these animals going.
So yeah, prepare to have your assumptions challenged, maybe, because this deep dive, I think, is going to uncover some remarkable connections, stuff linking the tiny world inside our cells to, the grand scale of lifespan across mammals.
Okay, let's unpack this.
Right, let's start with the big picture. So inside our cells, proteins are constantly being, broken down and rebuilt. This continuous process. That's what we call protein turnover.
A cycle, basically.
Yeah.
Degrade, synthesize, repeat.
Precisely. It's fundamental. In this study, it revealed a genuinely striking pattern across all the different mammals they looked at.
They found a negative correlation. So between lifespan and the turnover rate, specifically of the most abundant proteins.
Okay, negative correlation means longer life, slower turnover.
Exactly. Mammals that live longer tend to replace their most common cellular proteins more slowly.
And you mentioned the range of lifespans was huge.
Oh, enormous. Like you said, maybe 4 years for a mouse, up to 200 plus for the bowhead whale. That's a massive difference.
Wow.
And this wasn't just some vague trend. They even found it when comparing the same proteins, the equivalent ones, or orthologous proteins as they're called, across these different species.
So like comparing the same engine part in a mouse car versus a whale car, kind of?
Huh. Yeah, that's a decent analogy. The function is conserved, even if the overall vehicle is very different.
Interesting. And what about body size? Because you'd think bigger animals might live longer usually.
They did look at that, yeah.
Yeah.
And there is a correlation with body mass, but the link they found between lifespan and this protein turnover rate, that was actually stronger.
So turnover is a better predictor of lifespan than just size in this context.
It seems so, based on this data. And what's really interesting is that this relationship holds up regardless of how closely related the species are evolutionarily.
Right. Okay.
Which points toward something pretty fundamental going on, a core biological principle.
Okay, so longer life, slower turnover for these common proteins. Got it. But why? That's the big question, isn't it? Let's dig into that, the abundant proteins and the energy side of things.
Good question. The study really highlighted that this connection, this lifespan turnover link, it's particularly strong for the proteins that are, most numerous in the cell, the really abundant ones.
Okay.
And this starts to make sense when you remember that protein turnover isn't, free. It costs energy, a lot of cellular energy.
Right. It takes effort to break down and rebuild things constantly, like nonstop renovation, as I said earlier. It must be expensive energetically.
Exactly. So the researchers started thinking, could it be that for these really long-lived species, replacing these super common proteins more slowly is actually an energy-saving strategy?
Ah, okay. If you're going to live for 200 years, maybe you need to be more frugal with your cellular energy budget.
That's the hypothesis, yeah. Any way to economize on energy at the cellular level could provide a huge advantage over such long timescales. Makes evolutionary sense.
Now, this is where it gets really cool, I think. They brought in the naked mole rat.
Ah, yes, the superstar of aging research.
Right. For anyone who doesn't know, they're these little rodents, kind of mouse-sized, but they live way longer than mice. Decades sometimes.
Extraordinarily long for their size. And crucially for this study, they also have slow protein turnover rates. Fits the pattern.
Okay, so far so good. But here's the twist, the paradox, right?
Exactly. Despite this slow turnover, their cells are actually more resistant to stress caused by misfolded proteins compared to mouse cells.
Wait, hang on. That feels backwards. If you're replacing proteins slower, wouldn't you expect more junk, more misfolded stuff to build up and cause problems?
You absolutely would. That's the counterintuitive part.
Mm-hmm.
So they tested this. They used a chemical called AZC. It mimics an amino acid, proline, but it messes up proteins, causes misfolding when it gets built in.
No.
And they found naked mole rat cells could handle much higher doses of this AZC compared to mouse cells, at least when the cells weren't dividing.
So they're slow at replacing proteins generally, but better at dealing with the faulty ones. How does that work?
Well, the idea that emerged is that maybe long-lived species didn't just slow everything down. Maybe they evolved more sophisticated selective ways to deal with the damaged goods.
Not just throwing everything out quickly, but having a really good quality control system.
Exactly, like having a highly skilled repair crew that targets specific problems rather than just demolishing and rebuilding the whole block all the time, which is very energy intensive.
That makes more sense. Okay, so let's connect this back to energy use and those reactive oxygen species, ROS.
Right. So if slower turnover saves energy, that means less demand for ATP, the cell's main energy molecule.
Makes sense.
And the researchers checked this. They measured ATP production rates. And guess what? Naked mole rat cells make ATP at lower rates than mouse cells, both through glycolysis and the main powerhouse pathway, oxidative phosphorylation.
So lower energy production overall.
Yeah.
And making ATP, while essential, that process itself can create damaging byproducts, can't it?
That's the link to ROS, yes. Reactive oxygen species. They're like the exhaust fumes from the cell's energy production. And they can damage proteins, DNA, other cell parts.
And the naked mole rats.
They found lower levels of ROS in the naked mole rat cells, too.
Okay, so it's all fitting together now. Slower turnover, less energy needed, less ATP made potentially fewer damaging ROS byproducts. It's a whole chain reaction.
It seems to be, yeah. A potential benefit of that slower metabolic pace linked to the slower turnover.
Fascinating. Okay, but let's circle back to how they handle the misfolded proteins so well, despite the slow general turnover. You mentioned selective mechanisms.
Yes. The evidence really points to them being better at clearing out the specific proteins that are damaged. Remember that AZC experiment?
The stuff that causes misfolding.
Right. They tracked how much AZC actually got incorporated into proteins over time. Naked mole rat cells ended up with less AZC built into their proteins compared to mouse cells.
Which is good, obviously. Less faulty material.
It is. But here's the clever part. This difference wasn't uniform across all proteins. Some proteins showed a big difference in AZC incorporation, others less so.
So it wasn't just that they slowed down all protein building when AZC was around. It was more specific.
Exactly. That suggests some kind of selective process is happening. Maybe they're better at spotting and removing proteins that do incorporate AZC before too many accumulate.
Okay, so how do cells normally get rid of specific unwanted proteins?
The main system for that is the ubiquitin proteasome system, the UPS. Think of ubiquitin as a little tag.
A kick-me sign for proteins.
Sort of, yeah. It gets attached to proteins that are damaged or no longer needed. And that tag signals the proteasome, which is like the cell's recycling center or garbage disposal, to come and break that protein down.
Right, the UPS. So how does that play into the naked mole rat's resilience?
Well, they found that the naked mole rat's ability to tolerate that AZC chemical was completely dependent on having a working proteasome. If they blocked the proteasome with an inhibitor, the naked mole rat cells lost their advantage. They became sensitive to AZC, much like the mouse cells.
Wow. So that targeted disposal system, the proteasome, is absolutely critical for their tolerance, even with slow overall turnover.
It really seems to be. And they did another cool experiment. They looked at the levels of these ubiquitin-tagged proteins. Normally, AZC-treated naked mole rat cells had fewer tagged proteins floating around than mouse cells.
Suggesting they're clearing them out efficiently.
Exactly. But then when they blocked the proteasome, those tagged proteins piled up much faster in the naked mole rat cells.
So the tagging system is working, maybe even faster, but the disposal is usually so efficient you don't see the buildup.
That's the interpretation. It suggests a faster rate of clearance. They're identifying, tagging, and destroying the damaged goods more rapidly and efficiently, even if their general protein replacement is slow.
So the picture emerging is really quite elegant, isn't it? It's like these long-lived mammals, or at least the naked mole rat example, they save energy with slow general turnover.
Energy conservation.
But they compensate by having these hyper-efficient, targeted quality control systems, like the UPS, to deal with specific problems as they arise.
Precisely. It's a fascinating trade-off. Slow and steady for the bulk, but fast and precise for the cleanup.
That completely flips my initial thought. I assumed slow turnover just meant living with more potential damage, but it's about being smarter, more selective.
Yeah, it really reframes things. And these findings, they have potentially big implications for how we think about aging overall.
How?
Well, it suggests there isn't just one single strategy for longevity across all life. You mentioned, for instance, that some studies in invertebrates like worms or flies have sometimes linked longer lifespans to increased protein turnover.
Oh, the opposite.
Right, which suggests that different branches of life might have evolved fundamentally different solutions to the same problem, maintaining protein health over time.
So mammals might favor this energy-saving, selective cleanup approach, while maybe some invertebrates went for a more rapid, replace-everything strategy.
It could be. It highlights this core biological tension, this trade-off, between how much energy you spend constantly replacing proteins versus the need to make sure those proteins are working correctly.
And long-lived mammals seem to have tilted the balance toward saving energy, but being really good at targeted repairs.
That seems to be the story emerging from this line of research, yes. Especially for these highly abundant proteins where the energy cost of rapid turnover would be particularly high.
But hang on. If slower turnover saves energy and seems linked to longevity in mammals, why haven't all mammals evolved this? Are there downsides we're missing? Maybe faster turnover is better for responding quickly to infections or environmental changes in shorter-lived animals?
That's an excellent point and absolutely something to consider. Evolution is full of trade-offs. Faster turnover might confer advantages in certain contexts, maybe faster adaptation, repair after acute injury, or dealing with specific pathogens. This study highlights one successful strategy, particularly prominent in very long-lived mammals. But it doesn't mean it's the only strategy, or that it doesn't come with its own set of compromises.
Right, it's complex. Okay, so let's try and summarize the key takeaway from this deep dive. We started asking why slower protein turnover might link to longer life in some mammals. And the study suggests that, yes, for mammals, longer lifespans often correlate with slower replacement of the most common proteins. This likely helps conserve energy and might reduce damaging byproducts like ROS. But critically, as the naked mole rat shows us, this doesn't mean they just accumulate damage. Instead, they seem to have evolved these really efficient selective systems, like the UPS, to find and remove specific misfolded or damaged proteins very effectively.
Exactly. It's not just about slowing down. It's about combining that energy saving with enhanced targeted quality control.
So it's a strategy of being frugal with general maintenance, but investing heavily in specialized repair crews.
A great way to put it.
Which leads us to the final thought for you, our listeners, to chew on. If this balance between slow general turnover and efficient targeted clearance is key for longevity in some mammals,
could understanding that balance better, maybe even finding ways to influence it, be a pathway towards healthier aging in humans?
Yeah, what could this mean for human health research? For tackling age-related diseases where protein misfolding is a problem?
What are the implications if we could, say, boost the efficiency of our own cellular repair crews while maybe promoting energy efficiency elsewhere? Lots to think about.
Some truly fascinating food for thought indeed. Thanks for listening, and as always, don't forget to like the video and subscribe if you haven't already.